Vaccinium, corymbosum, anthocyanin concentration.
What you see here is a single fruit of the high-bush blueberry, Vaccinium corymbosum, a plant belonging to the heath family, the Ericaceae. Its diameter is about 12 millimeters, smaller than a marble. The entire documentary that follows takes place exclusively inside this one fruit. Botanically, it is an epigenous berry, also called a false berry. It does not arise from the ovary alone, but from an inferior ovary, in whose construction the floral cup, or hypanthium, and tissues of the sepals also take part. The edible mass is therefore partly floral tissue, not solely ovary wall. This fruit is, biologically speaking, an architectural composite of different tissues serving a single function: the reception, protection, and dispersal of seeds. In the course of this documentary, the camera will traverse the tissue layers of the fruit from the outside in, from the cuticle, through the flesh, and on to the seeds. In doing so, cellular and biochemical processes become visible that are normally hidden from the unaided eye. The respiration of individual cells, the deposition of pigments, the remodeling of the cell wall, the transport of sugars through the conducting tissue, and the hormonal control of ripening. The high-bush blueberry is the product of decades of breeding from wild Vaccinium species. The fruit examined here nonetheless represents the same fundamental biological processes that occur in wild forms. Cell division and cell expansion determine growth, osmotic processes determine turgor, and the phenotype of the ripe fruit, color, aroma, texture, is the outcome of coordinated genetic and biochemical programs. The chapters that follow treat these programs in the order in which the tissues are traversed, from the surface toward the center. The outermost layer of the fruit is the exocarp. It consists of the epidermis and several underlying subepidermal cell layers. Outward, the epidermis is sealed by the cuticle, a water-repellent layer made of cutin and waxes. In the ripe blueberry, this cuticle is remarkably thick. On the cuticle sits the epicuticular wax, which produces the so-called bloom of the blueberry. This wax forms crystalline structures in the shape of cylindrical rods and tubes about 1 micrometer long and roughly 200 nanometers in diameter. To picture the scale, a micrometer is 1/1000th of a millimeter, so several of these rods would fit across the width of a single human hair. These wax crystals are the reason for the frosted, bluish sheen of the surface. They reflect part of the incoming light, reduce the fruit's transpiration, and hinder the entry of microorganisms. Fun fact: that dusty-looking coating is not dust at all. It is a forest of microscopic wax crystals. If you rub a blueberry, and if you rub it quite hard, the bloom comes off and the fruit appears darker underneath, because you have removed a plant-produced protective wax layer, not a layer of dirt. In the epicarp, especially in the epidermis and the subepidermal layer, the pigments of the fruit reside. Moreover, contrary to older assumptions, the blueberry fruit does possess stomata, the small pores used for gas exchange, but they are hidden beneath the dense wax covering. The majority of the chlorophyll and the stomata are carried by the persistent sepals at the distal end of the fruit. In these tissues, particularly in unripe, green developmental stages, a process of photosynthesis and carbon dioxide recycling takes place. In the ripe, blue flesh, this process is not to be regarded as an active main activity. The cuticle is subject to remodeling throughout the entire ontogeny of the fruit. Its thickness and the density of the epicuticular wax crystals increase as fruit development proceeds. Cuticular waxes typically contain long-chain aliphatic compounds, such as alkanes, alcohols, esters, and fatty acids. Depending on species and developmental stage, other lipid classes may also be involved. These lipids are synthesized in the epidermal cells, transported through the cell membrane, and crystallized into structures at the surface. The barrier function of the cuticle is therefore not a static trait, but the outcome of a continuous, metabolically costly process that protects the fruit from water loss and from the entry of pathogens. Beneath the exocarp follows the mesocarp, which makes up the greater part of the edible fruit mass. In high-bush cultivars, the mesocarp can constitute about 4/5 of the ripe fruit tissue. In particular, the outer mesocarp is the decisive factor for the variation in fruit size and weight among different genotypes. The mesocarp consists of parenchyma cells. These cells are large, round, and nearly isodiametric. Their interior is occupied by a central vacuole that fills almost the entire cell volume. The cytoplasm is distributed as a parietal rim along the cell wall. This architecture is characteristic of storage tissues in fruit. The internal pressure of these cells, the turgor, arises from the osmotic influx of water into the vacuole. The turgor pressure pushes the cell membrane against the cell wall and gives the tissue its firmness. To picture this, each cell is like an inflated water balloon, firm and taut when full, limp and wrinkled when it loses water. If the cells lose water, turgor falls and the fruit loses its snap. A blueberry is therefore not a liquid-filled cavity, but a pressure-tensed assembly of cells. Scattered among the parenchyma are sclerides, the so-called stone cells. These cells have strongly thickened, lignified cell walls and serve as mechanical support elements. The degree of sclerification in the subepidermal layers correlates with the firmness of the fruit. More sclerified cultivars show higher textural firmness. To continue the analogy, if the parenchyma cells are like soft water balloons, the stone cells are like pebbles embedded among them, giving the tissue its slight grit. Between the parenchyma cells are intercellular spaces, gas-filled cavities whose surfaces may be coated with apoplastic fluid that serve gas exchange. Through these spaces, oxygen diffuses to the mitochondria and carbon dioxide from respiration is removed. The walls of neighboring cells are connected by plasmodesmata, channel-like structures that establish cytoplasmic connections between cells and enable symplastic transport of molecules. The fruit tissue is thus not an isolated collection of individual cells, but a communicating assembly whose cells exchange substances through plasmodesmata and through the cell wall apoplast. Fruit growth itself rests on two phases. A substantial portion of cell division begins before anthesis. After fertilization, cell expansion dominates, along with tissue-specific further cell divisions, which together determine the final fruit volume. The blue-violet coloration of the blueberry is not located in the cell walls, but in the vacuoles of the epidermal and subepidermal cells, in part within so-called anthocyanoplasts. The pigments are water-soluble flavonoids, termed anthocyanins. The anthocyanins of the blueberry are derived from 5 principal aglycone classes: delphinidin, cyanidin, petunidin, pianidin, and malvidin. In some reports, further aglycones, such as pelargonidin, are also mentioned. Each aglycone is linked to a sugar, most commonly glucose, galactose, or arabinose. In total, 15 to 25 different anthocyanin glycosides have been identified in the blueberry. The profiles are dominated by malvidin and delphinidin glycosides, with malvidin 3-O-galactoside frequently the most abundant individual anthocyanin. These figures, however, are species and cultivar dependent. The visible color of the fruit arises from the interplay of several factors: the pigment composition, the pH of the cell sap, complexation with metal ions, and the light scattering by the epicuticular wax bloom. The deeper flesh is not uniformly blue pigmented in all cultivars. In many cultivated varieties, the pigmentation is concentrated mainly in the skin and the outer layers. The anthocyanins are synthesized via the phenylpropanoid and flavonoid pathways, and are then sequestered into the vacuole by tonoplast transporters. The color is pH dependent. At lower pH, the pigments appear more reddish at higher pH more bluish. In their biological context, anthocyanins serve functions in protection against short wavelength radiation and in scavenging reactive oxygen species. In other words, they act as a kind of internal sunscreen and antioxidant shield for the cell. Fun fact: blueberries are among the comparatively few foods whose blue or blue-purple color is naturally occurring, rather than created by artificial coloring. Alongside the anthocyanins, the fruit contains further phenolic compounds arising from the same or related biosynthetic pathways. The principal phenolic acid of the blueberry is chlorogenic acid, an ester of caffeic acid and quinic acid. Additionally, caffeic acid, ferulic acid, and protocatachoic acid are present. Among the stilbenes, pterostilbene, a dimethyl ether analogue of resveratrol, and resveratrol itself have been detected, though in small, characteristic amounts. These compounds should be understood as plant secondary metabolites, fulfilling ecological functions, for example in defense against pathogens and in oxidative protection. The total anthocyanin content of the ripe fruit is reported in the literature at roughly 80 to 300 milligrams per 100 grams of fresh weight, with strongly pigmented cultivars reaching higher values. These figures are cultivar, cultivation, and method dependent. Through the pericarp run vascular bundles that connect the fruit to the parent bush. Each bundle consists of a sclerenchymatic sheath and a central strand of xylem and phloem. Fibers are associated with the conducting tissue, lending additional mechanical strength. The phloem transports assimilates, chiefly sucrose, from the photosynthetically active parts of the plant into the fruit. The xylem conducts water and dissolved minerals, especially in early developmental stages. Transport proceeds along concentration gradients and, in the case of the xylem, through transpiration-driven tension. There is no active circulation comparable to the animal bloodstream. There is no central pump, only gradients, diffusion, and evaporation. In the ripe blueberry, glucose and fructose together make up about 10% of the composition. The fruit is a metabolic sink. It stores the sugars and acids supplied by the bush and remodels them during ripening. The sugar is stored partly in the vacuole and partly converted into other compounds. For the fruit to grow, the bush must supply it continuously. The fruit itself is not self-sufficient. In the xylem, water transport is maintained by the cohesion of water molecules and by transpiration-driven tension. In the phloem, assimilate transport follows the pressure flow model. In the source tissues, the photosynthetically active leaves, sucrose is loaded into the sieve tubes. The resulting osmotic influx of water generates a hydrostatic pressure that drives the sieve tube contents toward the sinks, here, the fruit. In the fruit, sucrose is unloaded and split into glucose and fructose, or converted into other storage forms. This is why a blueberry left on the bush can keep gaining sugar and get sweeter, but a blueberry picked too early cannot import any more sugar through the phloem. Any apparent change in sweetness after picking is then due to water loss or internal metabolic shifts, not to the fruit gaining sugar. Every cell of the fruit is alive and carries out cellular respiration. In the mitochondria, simplified, sugar is oxidized under consumption of oxygen to yield energy in the form of adenosine triphosphate, or ATP. The end products of this respiration are carbon dioxide and water. This process continues in every ripe fruit cell, even after the fruit has been separated from the bush, for as long as the cells remain alive. A blueberry, after harvest, is still breathing. Its cells consume their own stored sugars and oxygen and release carbon dioxide. This is why fruit kept too long at room temperature loses sweetness and firmness. It is, in a sense, consuming itself. Alongside respiration, the fruit cells store organic acids. The predominant organic acid in the blueberry is citric acid. In many studies, it accounts for between 60 and 80% of total acids, though this share is cultivar, ripeness, and method dependent. Further organic acids include malic acid, quinic acid, shikimic acid, and succinic acid. These acids are not a waste product of respiration, but stored metabolites forming the acidic counterpart to sugar. From the ratio of sugar to acid arises the taste of the ripe fruit. During ripening, the acid content falls while the sugar content rises. The fruit becomes sweeter and loses acidity. Citric acid and the amino acid phenylalanine correlate positively with fruit firmness. The organic acids also serve as intermediates in metabolism and as reserve substances of the cell. The citric acid itself is an intermediate of the citric acid cycle, the central oxidative breakdown pathway of cellular respiration that takes place in the mitochondria. In the fruit, however, a portion of this metabolite is not further processed, but is accumulated in the vacuole. This accumulation is an energy-dependent process. The acids are moved into the vacuole via tonoplast transport processes and proton gradients and accumulated there, where they contribute to the osmotic system of the cell and at the same time determine the pH of the cell sap. From the interplay of sugar, acid, volatile aroma compounds, and texture arises the overall sensory impression of the ripe fruit. Additionally, the blueberry contains vitamin C in the form of ascorbic acid, dietary fiber, especially in the form of pectin, and the minerals potassium, phosphorus, and magnesium. Ripening of the blueberry is a coordinated process in which color, texture, and sugar and acid content are altered together. The fruit shifts from green through pink to blue-violet, a process accompanied by the synthesis of anthocyanins. The softening of the flesh rests on a controlled breakdown of cell wall polysaccharides. Several processes can be distinguished. The solubilization of pectin occurs mainly in early and intermediate ripening stages. The depolymerization of hemicellulose is detectable across all ripening stages. In addition, there is a loss of arabinose from the side chains of cell wall polysaccharides and a breakdown of the middle lamella that holds the cells together. To picture this, if the cells are like bricks, the middle lamella is the mortar between them, and during ripening, that mortar is gradually dissolved, so the bricks, the cells, can slide apart and the tissue softens. Various enzymes participate in these remodeling processes. Demonstrated for the blueberry are pectin methylesterase, polygalacturonase, beta-galactosidase, and cellulase. Across fruits in general, pectate lyases and expansins are also known as cell wall loosening agents. The interplay of these enzymes leads to a loosening of the polymer network, to a swelling of the primary cell wall, and thus to the softening of the fruit. Firmness is also determined by pectin-calcium dynamics. Demethyl-esterified pectins can form so-called egg box structures through calcium bridges, which mechanically stabilize the cell wall. The hormonal control of ripening is remarkable in the blueberry. The fruit shows during ripening an increase in respiration rate and in ethylene production. However, ethylene biosynthesis is not autocatalytic but developmentally regulated. For this reason, the blueberry is described as atypically climacteric. More recent work classifies it as non-climacteric, with ethylene-dependent, genotype-dependent ripening traits. A uniform classification as classically climacteric or as non-climacteric is not scientifically settled. Whether a blueberry counts as climacteric is a genuine, ongoing scientific debate. Climacteric fruits, like tomatoes and bananas, ripen with a burst of respiration and ethylene and can be ripened after harvest. Non-climacteric fruits, like strawberries, do not. The blueberry sits awkwardly between the two categories, and the answer depends on cultivar and on how climacteric is defined. Alongside ethylene, abscisic acid is another central regulator of ripening, governing coloration and anthocyanin accumulation. Ethylene experimentally accelerates softening by enhancing pectin breakdown and the activity of pectin methylesterase and polygalacturonase. Over the course of ripening, the composition of secondary metabolites also shifts. The anthocyanin content rises markedly with advancing ripeness, whereas the content of chlorogenic acid and other phenolic acids partially decreases. The color of the fruit, therefore, does not change abruptly, but in a continuous process in which, step by step, new anthocyanins are synthesized and deposited in the vacuoles. This coordination is mediated by transcription factors, in particular those of the MYB family, which regulate the expression of the genes of the flavonoid pathway. Ripening is thus not an isolated biochemical event, but a genetically programmed, hormonally controlled overall process in which metabolism, cell wall remodeling, and pigmentation proceed in synchrony. The fruit is not, functionally, an end in itself. It serves the protection and dispersal of the seeds. The blueberry is a multi-seeded berry arising from an inferior 4 to 5 locular ovary with axile placentation. The ovary may contain more than 100 ovules, from which, after fertilization, the seeds develop. Each ripe seed consists of the seed coat, the endosperm as nutritive tissue, and the embryo as the actual young plant. The embryo arises from the double fertilization of the flower. The endosperm supplies it with reserves during development and germination. Fun fact: bite a blueberry gently and you will notice a faint grit. The grit comes mainly from the tiny seeds, and in some cultivars may also be influenced by stone cells. A single berry may contain several dozen seeds, and each viable seed contains an embryo that can germinate under suitable conditions. The sweet, anthocyanin-rich flesh favors dispersal by fruit-eating animals. The seeds are ingested together with the flesh and can, after passage through the digestive tract, be excreted at a distant location, where they may germinate under suitable conditions. The fruit is thus a dispersal architecture. It invests sugar, pigments, and water in a tissue whose function is to attract a disperser and to transport the seeds. From the plant's perspective, the fruit-eating animal is a means of transport. The blueberry produces sweet flesh so that this flesh will be eaten. The seeds are the actual content to be deposited at a new location. In this sense, the entire preceding architecture of the fruit, cuticle, parenchyma, vacuoles, vascular bundles, is subordinated to the protection and the delivery of the seeds. Seed number and seed size vary among genotypes. As a rule, not every ovule develops into a ripe seed. Fertilization success, nutrient supply, and genetic factors determine how many seeds are ultimately formed. Each seed carries the genetic information for a new plant that may differ from the parent plant, since genetic recombination during sexual reproduction produces new gene combinations. The fruit is therefore not only a storage and protective organ, but also a vehicle of genetic diversity. Through seed dispersal by animals, the plant can reach new habitats and distribute its genes over larger distances than would be possible through vegetative reproduction alone. With the completion of the ripening phase, processes of senescence and tissue breakdown set in. The cell wall remodeling that began during ripening continues. The middle lamella is further dissolved, the cell walls lose cohesion, and the cells separate from one another. The membrane integrity of the cells is lost. The ordered interior of the cell becomes disordered. Over the course of senescence, metabolic activity typically declines. The extent and time course depend on genotype, ripeness stage, and storage conditions. The fruits become more susceptible to microbial attack. Microorganisms enter through microcracks, wounds, natural openings, or weakened regions of the cuticle. The fruit surface had been the primary barrier against the entry of microorganisms. With its loss, decomposition begins. What began as a pressure-tensed cell assembly ends as a dissolving tissue. The 12 mm planet loses its structure. What remains are the seeds, seed coat, endosperm, and embryo, which, if they reach a suitable location, can grow into a new plant that in turn produces flowers, is fertilized, and again yields a blue berry. The cycle that began inside this one fruit closes outside it, in a new fruit whose interior will be a world of its own. In ecological terms, the fruit has done its job once the seeds are mature. The sweet, costly flesh is, from the plant's perspective, a one-way delivery package, meant to be eaten, so that the seeds inside are carried away and given a chance at a new life. This concludes the life cycle of the fruit. What has been observed in this documentary is the temporal sequence within a single berry, from tissue formation through ripening to senescence. The processes described here, cellular respiration, osmotic regulation, secondary metabolism, cell wall remodeling, and hormonal control, are not restricted to this one fruit, but are characteristic of fleshy fruits in general. The blueberry is thus an exemplary object on which the general principles of plant fruit biology can be studied in a particularly compact form. A habitat 12 millimeters in diameter, within which the fundamental processes of plant metabolism unfold in their full complexity.